Hi friends, a very happy new year to you all. This will be the first videos which I'll upload in 2021. Let's see what the topic is, and the topic is the cutaneous circulation. Why do we have to describe the cutaneous circulation separately? Look, the circulatory tree, the vascular tree has the same arrangement, same organization everywhere. I mean, left ventricle pumps the blood into the aorta, then there are arteries, arterioles, metarterioles, capillaries, and then we have venules, veins, venae cavae and right side of the heart. But then looking at the specific organs, we have some special characteristics for the circulation in that particular organ or tissue. We have seen pulmonary circulation. There are zones of blood flow in the lungs. We have seen renal circulation, coronary circulation. They have their own special characteristics. And so is the case with cutaneous circulation, blood flow in the skin. It has some major distinctive features which make it unique as compared to the other circulations. So let's see what those unique features are and how the circulation is arranged, and then finally we will also see the application of it in terms of the triple response or triple reaction when the skin is stimulated, how the blood flow responds. So let's start. Now, first thing at the very outset is that generally the blood flow to an organ depends on the metabolic activity of that organ. Greater the metabolic rate for a particular organ, greater the blood flow into that organ. This is the case everywhere. I have discussed this hypothesis in some other video. It has been called as Bernays hypothesis, where increased metabolic activity increases the ATP breakdown, and increased ATP breakdown means formation of the ADP, then AMP, and finally adenosine remains. This adenosine leaks out of the cells and dilates the vessels. So blood flow will increase. Increased metabolic activity, increased blood flow. That's the case everywhere. That's how the blood flow behaves in all the organs. Now there are only two exceptions. There are two exceptions to this. The two exceptions are, one is kidney. I have described renal circulation in some previous video. What is the exception in the case of kidney? Renal circulation. Look, at all other organs, in all other organs, increased metabolic activity increases the blood flow to that organ. In the case of kidney, it is other way around. That is, increased blood flow increases the metabolic activity. Do you get the point? Everywhere in the body, metabolic activity determines the blood flow to that organ. In the kidney, blood flow determines the metabolic activity in the kidney. So that's exception. And why is that? You can refer to the video that I made on renal circulation. I have described it in detail why it happens. And the second exception is skin, the cutaneous circulation which we are about to describe. In the case of skin, blood flow does not depend on the metabolic requirement of the skin. Blood flow depends on the requirements of the body temperature regulation, right? It's unique. All other organs, the metabolic activity is going to regulate the blood flow. In the skin, it's not the metabolic activity, but it's requirement to regulate the temperature of the body. That will determine the blood flow. In fact, if you see, the blood flow far outweighs or exceeds the metabolic needs and nutritional requirement of the skin. The nutritional requirement and the metabolic activity of the skin would be very insignificant maybe, but the blood flow can be huge. So it far exceeds the nutritional requirement in the case of skin. Right. Let's see some factual things also, like skin is the largest organ of the body in terms of the surface area. And there are regional variations in the structure of the cutaneous circulation, cutaneous vasculature. I mean, from region to region, the blood flow in the skin may differ. Well, there are common factors, common features which we can see now in the figure. It's the dermis and then the superficial and deep dermis. So what you can see here is that arterioles, venules, note the point that they are running parallel to the skin. The first point to be noted is that they are running parallel to the skin. Normally, when an artery and arterioles, they are entering a tissue, they generally enter in a perpendicular fashion. But here, the blood flow is running parallel to the skin surface. So that's something unique. Arterioles run parallel to the surface of the skin. And the second point that you can note here is the countercurrent flow, countercurrent blood flow in the arteriole and venule. That's the other point. Yes, countercurrent blood flow means going in the opposite directions. You can see in the diagram. There are at least 4 places in the body where you see the countercurrent blood flow. Countercurrent flow of blood is seen in skin, as I mentioned now. Apart from that, in the intestinal villi, the GI tract, there is a countercurrent flow. Then testicular blood flow, again countercurrent type. And of course in the kidney, countercurrent mechanism for concentration of urine, vasa recta. So in the case of skin, in the case of even testicular blood flow, the countercurrent flow favors heat exchange among the vessels. So what happens is, let's say skin, there's a ambient temperature is cold. Let's imagine that. So the blood that is returning from the skin, the veins, venules, they will be carrying a cold blood, and that can decrease the temperature of the blood that is running in the arteriole, which is reaching up to the surface now. You get that point? So it favors the heat exchange because of that kind of a parallel or countercurrent type of flow in the arterioles and venules. To just complete the structure, let's see, there is an arcade of arterioles at the boundary of dermis. So at the boundary of dermis, there will be arterioles, and then a dermal papilla will be there where a loop of capillary will be made. Let's see, we have another diagram. Yes, here it is. You can see, well, epidermis does not have the blood flow. Then the dermis, it shows the dermal papilla where there is a capillary loop. So the arteriole breaks into met arteriole going beneath that papilla, and that met arteriole will give rise to this capillary loop. Means at the halfway turn, the loop has been made, and then it breaks into the venule, which will come back. It will break into the venous blood. So that's how the arrangement is. Of course, above that is the epidermis has been shown, which does not have the blood flow. Then we have the second feature that can be noted here. One is this, the structural arrangement, parallel running vessels and this U shape of bend from the met arterioles to the venules, the capillary loop in the dermal papillae. The second thing that you should note in the diagram is the arteriovenous anastomosis. Note in the diagram that the arterioles have given off the collateral which make this anastomosis with the venule. So arteriovenous anastomosis. And this kind of arteriovenous anastomosis are present not all over the skin, not all over the body, but in certain regions, in certain specialized regions, which we will see in some time now. But these are the two things which we can note structurally. Arteriovenous anastomosis and the countercurrent flow. Next, what is the rate of blood flow to the skin? 1 ml per 100 gram of skin per minute. That's a basal blood flow in the cutaneous circulation, but it can increase to up to as high as 100 to 150 ml per 100 gram of skin per minute. So let's see the characteristics of cutaneous circulation. First, we have already mentioned the amount of blood flow far exceeds the metabolic requirements and nutritional requirements of the skin. We have seen that already. And why is that? It's because blood flow is not dependent on the metabolic requirement. Blood flow is dependent on the requirement of the temperature regulation of the body. That governs the blood flow in the skin. Second feature is massive changes in the blood flow that can occur in response to the temperature changes. Ambient temperature change, there can be a massive change in the blood flow, as we saw just now, from 1 ml per 100 gram of tissue per minute to it can increase up to 150 ml per 100 gram of skin per minute. Third point, skin acts as a reservoir of blood. Yes, this is in addition to the splanchnic circulation. Splanchnic circulation in the abdomen, in the digestive tract, it's a reservoir of blood. Whenever another organ, and particularly a vital organ, needs an extra blood flow, splanchnic circulation will divert its blood flow there. Pulmonary circulation can act as a reservoir of blood. We have recently seen one such in the video of pulmonary circulation, because it accommodates large quantities of blood at any given point of time. So even that can act as a reservoir, and cutaneous circulation, whenever required, can act as a reservoir of blood. Next point is about the peripheral resistance. The cutaneous circulation can also contribute to the peripheral resistance. Which circulation is the major contributor to the peripheral resistance? It's the skeletal muscle circulation. The major contribution in the peripheral resistance to the blood flow comes from skeletal muscle blood vessels. And why is that? Quite obviously, because the muscle is a contractile tissue. So it keeps on contracting and thereby compressing the vessels within. And since the vessels get compressed or constricted, narrowed, it will offer the resistance to the blood flow, and that's a major contribution in the peripheral resistance. The other contributor, as I mentioned just now, is the cutaneous circulation. And why is that? That's because cutaneous circulation, the vessels keep on constricting as per the lowering of ambient temperature, and because they keep on constricting, there would be a resistance to the blood flow. So that's another feature worth noting, that cutaneous circulation contributes in the peripheral resistance. Next, in the context of blood flow, the skin is of two types. Why am I dividing the skin like that? Just because the blood flow differs in these two regions which I am going to mention just now. One type is called as apical skin. So called apical skin because it's in the apical regions, apical skin. So earlobes, nose, lips, hands and feet, this skin in these regions is called as apical skin. You will very often notice that when you are in the very low ambient temperature, sub-zero temperatures, we feel differently for the nose or ear lobes. Sometimes you may have used this expression, I don't feel my nose at all. So yes, the reason is circulation is different in these regions. These are called, the skin is called as apical skin, and the other areas, which is actually the major part, is the non-apical skin. Now the cutaneous circulation, which shows numerous AV anastomosis, which we saw in the diagram, that is in the case of vascular circuit of the apical skin. So that one thing should be noted. And yes, one more point, these arteriovenous anastomosis, which are seen in the apical skin, they are called as glomus bodies. Yes, these are called as glomus bodies, but they are not related to those glomus cells. If you remember, the peripheral chemoreceptors present in the carotid and aortic bodies, the cell type is called as glomus cell. And these AV anastomosis in the skin are called as glomus bodies. They are not related. Only the name, the common word glomus is there, but they are not related. Anyways, but just remember glomus bodies, the arteriovenous anastomosis. Okay. Next point, countercurrent flow. We saw that just now, some time back, about the countercurrent flow. The arteries supplying the limbs have the accompanying veins coming back from that region. They are called as venae-comites, those veins. And these serve as the countercurrent exchange mechanism. There can be heat exchange. So this arrangement favors the heat exchange. Like as I said, in the cold environments, the cooled blood returning from the skin helps to lower the blood in the arterioles which is reaching the skin. So this is how heat exchange is possible. The arterioles which are taking the blood to the surface, and the veins which are coming back from the surface. Now because surface was cold because of the ambient temperature which is very, very cold, so this temperature or this blood in the veins can lower the temperature of the arteriolar blood which is going to the surface. And this is how blood temperature or core temperature can be regulated by the exchange of heat or regulating the transfer of heat. Right. Having said this, now let us see the factors that influence the cutaneous circulation or cutaneous blood flow. Two factors. We have already seen this at the start, that everywhere else in the body, the blood flow is regulated by the metabolic activity of that tissue or organ. In the skin, that's not the case. In fact, I told you, in the skin, the blood flow far exceeds the metabolic activity and nutritional requirement. So then, what are the factors that govern the blood flow, regulate the blood flow? Two factors. One is the neural regulation and the other one is the temperature, the ambient temperature and therefore the temperature of the skin. So let's look at these factors one by one. First, the neural regulation of the cutaneous blood flow. As we mentioned, the local metabolism, that's not the factor for the cutaneous blood flow, it's the nerves, the neural regulation. So, there is a slight difference which you should note now. How is it neurally regulated, the cutaneous blood flow? In the apical skin, it's different, and non-apical skin, it's different. In the apical skin, the anastomosis, they are under neural control. And look how it is. Because why you should know this is because they play a crucial role in temperature regulation, those arteriovenous anastomosis. So what happens is, decrease in the core temperature. I have mentioned this in the temperature regulation video. The skin temperature is the shell temperature and the core temperature is the temperature within, the temperature inside, the tissues and organs temperature. So if there is a decrease in the core temperature, then there will be a sympathetic discharge. And the sympathetic discharge releases norepinephrine. So there will be vasoconstriction in those anastomotic channels. Vasoconstriction in the anastomotic channels in the apical regions as nose, ear lobes, etc. And this will minimize the heat loss from the body. We need to conserve the body heat because core temperature had dropped. We need to maintain it. So vasoconstriction in those apical regions, apical skin, and particularly those anastomotic channels. So blood reaching the skin itself will decrease, and the heat loss from the skin to the surroundings will also minimize, and heat will be conserved in the body. But when the core temperature rises, then what happens? There will be simply withdrawal of that sympathetic tone. Do you understand this? There was sympathetic firing which caused vasoconstriction in the skin. When we have to lose heat, then simply that sympathetic tone will be withdrawn, so those AV anastomoses will dilate. There will be dilation in the cutaneous blood flow, in the cutaneous vessels. Which means what I am trying to establish here is that in those regions, there are no active vasodilator nerve fibers. There is no active vasodilation in those regions of the skin. There are vasoconstrictor fibers, they are released, sympathetic vasoconstrictor fibers, they cause vasoconstriction, and when their firing is stopped, the vessels will dilate. That's it. But now listen to this. In the non-apical skin, you will find there are active vasodilator fibers as well. So what happens is, vasoconstriction is brought about by sympathetic fibers that release norepinephrine. That's known to us. Now look at this carefully. Listen to this part carefully. There are active vasodilator fibers in the other areas of the skin, non-apical skin, and these are, listen to this carefully, these are sympathetic cholinergic fibers. Yes, normally we are aware that the sympathetic postganglionic fibers reaching any organ, they release norepinephrine as the neurotransmitter, and the parasympathetic postganglionic fibers reaching the organ releases they release acetylcholine. They are cholinergic fiber. Parasympathetic fibers cholinergic, sympathetic fibers adrenergic, releasing noradrenaline. Here we have sympathetic cholinergic fibers ending in the skin. They are releasing acetylcholine. So these are active vasodilator fibers, means they will cause active vasodilation, not just by withdrawal of sympathetic tone and therefore causing vasodilation. No, active vasodilation. So how is that? How do they achieve this? It's by either of the mechanisms. One is either by stimulation of sweat glands in the skin, and therefore secretion of sweat and locally released molecules. Vasodilator molecules are released locally because of the release of sweat, or co-release of vasodilator neurotransmitters. I mean in the same vesicles, or along with the release of the acetylcholine, there can be also release of CGRP, calcitonin gene related peptide, or VIP, vasoactive intestinal polypeptide. So they are co-localized along with the acetylcholine or along with the norepinephrine also at some other places. The neurotransmitters can be co-localized. So there can be release of these neurotransmitters, and that can bring about active vasodilation. Very interesting fact that makes a distinction between apical skin and non-apical skin, how it differs in the blood flow and changes in blood flow. Right, so that was one, the neural influences that regulate the cutaneous blood vessels. The second one, of course, which I mentioned, the temperature. The ambient temperature has a direct influence on the cutaneous blood flow. So second factor should also be noted, that it's not the metabolism that is regulating the blood flow. Having said all this, now finally let's see the applied aspect, and that's in the triple response or triple reaction. So whenever there is a mechanical stimulus applied to some area of the skin, there is some local response shown by the cutaneous vessels. Let's see what that local response is. When a sharp object is drawn over the skin, what happens is, let's say initially, to understand, that object was drawn mildly. We will see a white reaction. A white line appears within next 10 to 15 seconds in that area, along that line where the object was drawn. And this is called as white reaction. The mechanical stimulus, it caused contraction of the precapillary sphincter. You know why the area appears white is because, let's say, here is a capillary and a precapillary sphincter. There is a contraction of precapillary sphincter, and the blood was emptied from the capillary. So new blood is not coming in and blood was emptied from the capillary. Naturally, if there is no blood, the area will appear white. That's the reason for that white reaction. Well, but then this happened when the area was stimulated mildly. Now let's see what happens if the area is stimulated bluntly and with more force. Forcefully, an object, sharp object, sharp object was applied, or a line was created on the skin with a sharp object drawn along the skin. There will be a triple response, triple reaction. When the skin is stroked more forcefully with a sharp instrument, it will result in a local vascular response, and it's called a triple response. So what it is, it consists of three things, of course: red reaction, flare, and wheal. Let's look at them one by one. First, the red reaction. Now instead of white line, there would be a red line along that area of stimulus. A band of redness appears in about 10 seconds after the stimulus, and it's due to the local capillary dilation. You know, because of the mechanical stimulus, there was a local release of a vasodilator, generally the histamine, and that will cause a local capillary dilation. And that local capillary dilation will lead to that redness along the line of stimulus. Slapping on the cheek, you might have observed that after this, the cheek is slapped, there is actually the red reaction, the redness appears. So, first things first, remember it's because of the local capillary dilation, and it is independent of innervation. Remember this, it's possibly due to release of the local vasodilator like histamine. But the next reaction depends on the neural influence. Let's see how. Flare. Now there's going to be a flare up. The first reaction, that was red reaction, was confined to that region where the line was drawn. Now there the redness will flare and spread in the adjoining areas. So it's called flare. The redness does not remain restricted along the stroke line, it spreads out, flares up. This occurs because of the arteriolar dilation. So you must note it now. First reaction, the redness was because of the capillary dilation, therefore redness appeared. Well, white reaction was because capillaries were emptied, and that was because of contraction of precapillary sphincters. Then red line because of capillary dilation and increased blood flow. Now there is dilation of the arterioles. which cause flare. Let's try to understand this. The flare is mediated by axon reflex. The axon reflex, which I have described in the previous video on the axonal conduction, where I have described certain terms like antidromic versus orthodromic, anterograde, retrograde, unidirectional, bidirectional. So in that context, I mentioned that there can be antidromic conduction in the nerves. Look at this. This is about the antidromic conduction. Flare is mediated by axonal axon reflex. Look, an area of the skin shown in the figure, this area was stimulated, and what you can see is the afferent nerve, cutaneous afferent nerve, which is emerging from the skin and going to the spinal cord. Now, this area is stimulated, one area stimulated. The signals are carried by the afferent nerve. The signals are sent to the spinal cord. But then, what you can observe is the impulses are coming back in the opposite direction in the other branch of that nerve. This is an afferent nerve. Afferent nerve goes from periphery to the center, to the CNS. Here, what we can observe is afferent nerve, its branches carrying the impulse, but from the branching point, the impulse comes back, and there is a neighboring nearby arteriole which will be dilated by this impulse which has come in an antidromic fashion. This will be called as antidromic because afferent nerve, its branch, is conducting the impulse from center to the periphery, in that sense, not center to the periphery but toward the peripheral part, in that sense antidromic. And because this conduction has happened, the nearby arteriole will dilate and the redness will spread in the adjoining areas. That's called as flare, the flare up. So, the branches in which there was antidromic conduction, they release the vasodilator neurotransmitters and there is dilation of those arterioles. CGRP, we have already mentioned, calcitonin gene related peptide, it's a vasodilator, and substance P. These two will dilate the arteriole in the region, in the neighboring region, and it will be a flare up of that redness. So, what is the evidence of this which I said just now? Flare persists even after total sympathectomy. So, even after total sympathectomy, these three reactions, the flare, the second one, will still be happening, it will be visible. It means that the flare is not mediated by the sympathetic nerves to the cutaneous vessels. Fine. However, it is abolished with local anesthesia in the region. So, there was involvement of sensory nerves, definitely. That's why it will be abolished when there is local anesthesia in the region, or with sectioning of the sensory nerve from the region. If you cut the sensory nerve, somatic sensory nerve from the region, then it will be abolished, but not with sympathectomy. So, what does it tell us? That sympathetic nerve was not involved in this, but the local cutaneous afferent nerves were involved. Right. So, sectioning of the sensory nerve beyond the branching point will eliminate the awareness of the stimulus, as impulses will not reach the CNS, but flare reaction still persists. So, it goes to prove that flare was mediated by this axon reflex involving that cutaneous sensory neuron. As you can see here, if you section this nerve beyond the branching point, let's say here, if the nerve is sectioned, what will happen? Skin was stimulated peripherally, its sensation will not reach the cortex, but flare will still persist. So, that goes to show that flare happens because of the axon reflex with antidromic conduction. It was just an evidence that it's mediated by antidromic conduction and axon reflex. And finally, the third reaction in the triple response is wheal. A localized edematous swelling develops along the line of stroking. So, you might have observed it sometime that if you stimulate the area, there is first red line, then flare up, and finally the wheal, raised elevated area of the skin appears. It's due to the increased capillary permeability. So, in the region the capillary permeability will increase. So, edema fluid or fluid leaked out of the capillaries, extravasation of fluid will result in this wheal. So, that's the involvement of cutaneous circulation in the triple reaction or triple response. And that was, but when you discuss this, one must remember initial mild white reaction was because of contraction of precapillary sphincters. Then red reaction was because of capillary dilation, and flare is because of arteriolar dilation. Arteriole dilate in the nearby region. So, this clarity must be there. And finally, wheal is because of increased capillary permeability and extravasation of fluid leaking out of the capillaries. Well, that was a summary of cutaneous circulation. Once again, Happy New Year to you all. Let's look at a very happy, prosperous and healthy 2021 ahead of us. Thank you.